Nanofabrication. Please, Arnon.
Okay, so I'm Arnon. I'd like to thank the organizer for giving me the opportunity to speak here. I'll talk about the SP1 protein, a topic a bit changed from the other topics till now. SP1 protein is a novel scaffold building block for self-assembly nanofabrication. So stable protein 1, or as we call it, SP1, was isolated in our lab for populus tremula trees, and we found it to be an extremely stable protein. The SP1 extreme stability is demonstrated by its boiling stability, protease resistance, wide pH range tolerance, and stability in detergent, organic solvents, and chaotropic agents. This is the three-dimensional structure of the protein. Here you can see the protein dodecamer. This protein is self-assembled from 12 identical monomers. And this ring structure, you can see that the end terminals of the monomers face the inner pore of the protein. Those dodecamer rings can be also observed by TEM. Here you can see indicated by the white arrows the SP1 protein rings, and at the bottom the single SP1 dodecamer ring with its 11 nanometer diameter from side to side and 2 nanometer in the inner pore. So after you all know our key player, I would like to show you a few things we can do with SP1 and use it as a molecular scaffold for nanofabrication. First is this two-dimensional array of SP1. Here, using Langmuir blodgett, we can actually squeeze the protein to a highly ordered two-dimensional array. And you can see these hive-like hexagonal arrays of the SP1 rings on the surface. And those, again, are AFM images of the same surface. And you can see we achieve a nice micron-scale monolayer, where when we scratch the monolayer here at this right picture, you can see that the single proteins pop out of the surface, meaning that this monolayer is indeed composed of those single SP1 particles. So now that we can actually array this protein in a highly ordered array, we want to actually bind it to, selectively bind it to a target surface. We used to work on silica. Here you can see the mutant SP1 with the silica binding peptides painted gold in the inner pore of the protein. The left picture is when the peptides are confined to the inner pore, and on the right, where the peptides are exposed to the outer solvent. So this protein was produced in E. coli, and when we try to put it on silica surfaces, and those are AFM flooding topography images, where the surface is blue and the object on the surface appear brown, you can see on the right, this is the silica binding SP1 that binds quite poorly to the silica surface compared to other SP1 mutants. You can see that all of them has a quite poor binding to the silica surface. At this point, we introduced a chaotropic agent. Chaotropic agents are materials that mostly denature most of the proteins, but since we are working with a very stable protein, it only gives it an amount of flexibility. So by adding guanidine hydrochloride to the solution, we can actually take out the silica binding peptides, and you can see here on the right in this picture where we have an almost full surface coverage of the protein, while the non-specific binding of the other mutant is dramatically reduced. So we can actually controllably bind this protein to silica surface. We can do the same manipulation also on gold surfaces, and we can order it in a highly ordered array. The next thing was to bind nanoparticles to SP1, and we continued with the same mutant, the one that binds silica, now using a 5 nanometer silica nanoparticles. Here you can see an illustration of the protein with the silica nanoparticle and AFM topography images. Here on the left, this is the free protein, and in this picture in the middle, you can actually see a population of a protein in the height of 2 nanometer and the hybrid protein in the height of 6 nanometer that presents the silica nanoparticles. Using the same technique of AFM flooding topography, you can see the single particles here. Those are the green dots, and with the red dot is a silica nanoparticle on the SP1 protein on the silica surface. So we can take ready-made silica nanoparticles and bind them to the inner pore of SP1. But we wanted to do it also in another approach, and this is through biometallization. we actually took another mutant of SP1, one that presents histidine amino acid in the inner pore, and then reduced palladium ions in solution in the presence of the protein. In those 3 tubes, you can actually see here on the right, this is the clear brown solution that we get when we reduce palladium ions in the presence of this mutant. When we do the same procedure in the presence of the wild-type SP1, without those histidine amino acid, we get this black precipitate, meaning that here in the brown solution, we actually have nanoparticles that are quite dispersed in solution. We can also get nanotubes, and I will soon show you how those palladium nanoparticles nucleated in the inner pore of SP1 can serve as connector between adjacent protein units. When we take for this tube number 4 a sample and view it using TEM analysis, you can see here we have nanotubes. Those nanotubes are actually made from palladium nanoparticles that are metallized in the inner pore of the protein and actually serve as connector between adjacent proteins. So we have a protein, palladium, one after the other, and we get those nice nanotubes. So actually we have 2 methods of introducing nanoparticles to the inner pore of SP1. We can also controllably bind this protein to surfaces and also control its highly ordered array, its highly ordered 2-dimensional array. Next thing is to use SP1 in order to present catalytic domains. Here we used the enzyme called glucose oxidase. This enzyme is a highly important enzyme in the biotechnology industry. It is used in fermentation procedures. It is used in biosensors and also in the research of biofuel cell. We took this glucose oxidase and actually genetically fused it to SP1 in order to use the SP1 ring as a scaffold to present the catalytic domain of glucose oxidase. Those are the TEM images that we got. Here you can see in the middle, this is SP1 with its 11 nanometer diameter from side to side, and it is encircled by 1, 2, 3, up to 12 units of glucose oxidase that go all around and are presented on the SP1 scaffold. We can also use the inherent virtue of SP1 to form those nice tubes. And you can see here that when SP1 forms those tubes, we actually have SP1 in the middle as an axis and the glucose oxidase SP1 and the glucose oxidase enzyme that encircle this protein all around. So this was relatively quite nice when we saw it, of course, but we wanted to use all the other techniques that I've showed you to introduce nanoparticles into this particle in order to harness the enzymatic activity of glucose oxidase and harness the reduction of glucose to extract those electrons here. So we actually took this protein that can bind to gold nanoparticles and also can present glucose oxidase, and in collaboration with the group of Professor Mazzei in University of Rome, we introduced this protein to gold surfaces that are modified with gold nanoparticles. So now we actually have a monomer on the surface of SP1s that bind to gold nanoparticles and present the glucose oxidase enzyme. So the next step was to actually measure the cyclic voltammograms that correspond to the biocatalytic of glucose by those hybrids. And you can see that as the glucose concentration goes up, we have more and more current and more cyclic voltammograms that correspond to this biocatalytic oxidation. So we actually have a monolayer, but I told you that we wanted to utilize these big particles, these big nanotubes, enzymatic particles. So we go on and put more and more protein on the surface. And here on the left, you can actually see the SPR signal indicating 7 to 8 layers of proteins on the surface. All of the proteins actually present gold nanoparticles in the inner pore. So we have those nice towers of enzymes, nanoparticles embedded in the inner pore of SP1, and glucose oxidase presented all around this tower. But you can actually see that we do not have any connection between those gold nanoparticles, between the enzyme. We need to connect them somehow in order to harness all the enzymatic activity of all the enzyme and sense it on the electrode. Here we actually use the glucose oxidase reaction. Glucose oxidase produces hydrogen peroxide, This can be used to reduce gold salt and enlarge the gold nanoparticles in the inner pore of the protein. And as we go on and do this reaction, we actually can achieve a nanowire inside the tube. And you can see here in the course of time how we get more and more current. And at approximately 40 minutes, we can actually reach a plateau where we have a positive connection of all the nanoparticles inside this tube. And again, the cyclic voltammograms of this setup corresponding to the biocatalytic oxidation of glucose. And you can actually see that as we give more and more glucose to the setup, we have more and more current. And the last thing I would like to show you is a comparison of the monolayer to this enzymatic nanotower wired to the surface. And you can actually see that the wired enzyme tube demonstrates 23-fold higher current than the monolayer, meaning that when we build an enzymatic tower and wire all the enzymes to the surface, we can actually achieve a 23 higher current. So at this point, I'll have to conclude. We have used SP1 successfully as a molecular scaffold for nanofabrication. This protein can orient nanoparticles. It can assemble 2- and 3-dimensional structure. We can selectively cover various surfaces. I've shown you silica. We can do the same with gold. We can controllably expose binding functionalities that are embedded in the inner pore of the protein. This protein can display catalytic domain. I showed you glucose oxidase, and we've done it with other protein domains, such as the cohesin-dockerin interaction, if you know. And last thing, the SP1 can serve as a molecular building block for future devices. I would like to thank all my group members. The group is big, so I won't go through all of them, but for my collaborators, Dr. Dani Porat in Givat Ram, Dr. Gil Markovich here from Tel Aviv University, Silke Burns in Germany, and the group of Professor Matzei in the University of Rome. Thank you very much.
Thank you very much, Arnon, for this beautiful talk. Any questions or comments? If there are no further questions or comments, thank you very much, Arnon.